Hey, it's Brett again. I'm here with the Module 3 lecture video. Today's probably going to be a short one because I also have a guest lecture lined up. Okay, a YouTube video, which says things better than I can say. So with that, let's get started. Share my screen. All right. So what we're working on for Module 3 is biological molecules. So this is kind of a continuation of our discussion of chemistry and how it applies to living organisms. And all these foods that you see here are packed full of 4 different types of biological molecules. And there's probably thousands, if not hundreds of thousands of different molecules in the items that you see here, but they fall into these 4 main categories that we'll be talking about. And then there's maybe even a bigger picture pattern that we'll try and look at as well. Okay, so the big ideas from this chapter. Why carbon? Carbon is kind of the atom that is most associated with biological molecules and living organisms. We call ourselves carbon-based life forms, but why? So we'll try and answer that question. We'll talk about this idea of monomers and polymers. And then most of the classes of biological molecules, most of those are going to be covered by the guest lecture video. So carbohydrates and lipids and proteins, I'm not going to say much about today. We'll talk a little bit more about nucleic acids, because that doesn't come through in the lecture video very well. Yeah, so again, I'll try and keep this short and to the point. So the thought for the day, you are what you eat. Well, sort of. Everything that makes up your body, you at one point ate, right? But not everything of what you eat is kept and retained in your body. Some things you use for energy, some things you expel as waste products, and some things you hold on to. So we'll talk about the different types of molecules and what they might, what their fates might be in your body. Okay, so here is the periodic table as seen by an organic chemist. I think I've already shown this to you at least once. But the big star of the show here is carbon. Carbon is the special atom kind of in the, in the second row, basically in the middle of the periodic table. Why carbon? Why is it so important? Well, we already saw an example of how carbon bonds with other atoms, and that was in methane. So carbon can share electrons with 4 other atoms. In this case, it's just 4 other hydrogen atoms. So this is really the simplest organic molecule containing carbon and hydrogen. And each carbon, carbon has 4 electrons in its outer shell, the blue ones here. And each hydrogen atom only has 1 electron in its outer shell, and they're going to bond so that they fill up that outer shell. And what this means is that carbon needs to bond, carbon needs to form 4 other bonds. With hydrogen, it's just 4 single covalent bonds, and these happen to be nonpolar covalent bonds, if we remember from Module 2. But carbon can bond with a wide variety of other atoms. So it can bond with oxygen, it can bond with other carbon atoms, it can bond with nitrogen. And in doing so, it can create a huge diversity of molecules. So here's an example of a molecule that you maybe are familiar with. This is caffeine. And you can see there's some carbon in here, carbon here, carbon here. Well, where's all this other carbon? It looks like there's a lot of nitrogen and oxygen. Well, it turns out in a lot of these diagrams for organic molecules, the carbons just aren't labeled because laziness, I think. So we're gonna go in and label the carbon atoms. We'll do this in green here. That isn't really showing up very well. We'll make the line a little thicker. Oh no, that's the highlighter. Let's clear all those. Okay. We'll draw the lines, format. Oh, there we go. Line width. Okay. There we go. Let's put a carbon there and a carbon there. Basically, anywhere there's a corner that isn't labeled, there's a carbon. And if there's only one line, that means we have a single bond, like between this carbon and this nitrogen, there's just a single bond. But if there are two lines, that means a double bond. So oxygen can form two bonds, and carbon can use two of its four bonding spots to bond with oxygen with this double bond. So that's kind of what's going on here. The other thing that is not labeled, sometimes if there's, like this carbon only has 3 of the 4 bonds filled. So actually there is an assumed hydrogen atom. Anytime you count up the bonds and there's not quite enough, you can assume there's a hydrogen. So, okay, so that's kind of a little bit of how to decode these organic molecules and make sense of them. But you don't need to really memorize this, just to know that carbon is kind of everywhere in all the molecules that we are made of, and also a lot of the molecules that are interesting to us, we'll say, like caffeine, right? That obviously has some physiological effects based on how it interacts with our biological molecules. And we'll get into how that works probably in module 4, or actually probably, I think module 5, we'll start to talk about enzymes and proteins and how small molecules can fit into enzymes. So, okay. So we look at this periodic table with the electron arrangements, and we can actually go through and say, oh, okay, we can figure out how many bonds these different molecules are going to form. And it turns out everything in the first column, well, hydrogen can form 1 bond. Everything in the right column forms 0 because the electron shells are already full. The atoms over here tend to give up their outermost electron pretty easily and become positively charged ions with a +1 charge. Carbon, we just talked about, it's in the middle. It can form 4 bonds. Nitrogen has 3 empty spots in its outer shell, so it can form 3. Oxygen can form 2. And fluorine and chlorine, those tend to actually form negatively charged ions because they're so close to having that full outer shell. What else? Phosphorus can form 3 or sometimes 5. It's a little bit of a weird one. I'm not going to get into all the reasons. Sulfur can form 2. Boron, it's got 5 open spots, but it's probably not going to fill all of those. So I'm going to say maybe 3 ? But it's never going to be a really happy atom with a full outer shell, just because there's too many electrons it needs to hold on to, and it's got too many in its outer shell to get rid of them all and form a positively charged ion. In the second row, magnesium likes to form an ion. Beryllium, I don't know. I don't actually know what beryllium does. Talk to a chemist if you're really curious about that. So anyway, that's kind of the general pattern for bonds. And as we can see, carbon can basically form the most. If we ignore kind of the weird behavior of phosphorus, carbon is it. And it turns out a lot of science fiction writers will say, oh, but maybe there's silicon-based life forms, and those could also form for bonds with other atoms. So there's a lot of silicon on Earth, but it's usually bound up to rocks in the form, or bound up to oxygen in the form of silicon dioxide, which is very common in rocks. So anyhow, that is the short crash course of chemistry and bonding behaviors of the periodic table. And carbon is basically the best at bonding with other things. Okay, so that's why carbon, right? So now we're going to get into the idea of monomers and polymers. Before we do that, we should talk about word roots a little bit. Mono means one, poly means many, di means two, oligo means a few, and mers means parts. So monomer just means one part, and polymer means many parts. And the basic idea for biological molecules is that we have these small building blocks that we call monomers, and we can link them together to form really big molecules called polymers. So if these little arrow-shaped things are our monomers, these are small organic molecule subunits, and they have kind of these attachment points, and they can get linked together to form a polymer. So a polymer is just, you take one monomer, add it to another, and then keep on adding monomers, and eventually you can get hundreds or thousands of monomers linked together into a polymer. Actually, I should say even millions for the largest of the polymers. So, okay. So how do these polymers form, and how do they split apart? The formation of a polymer in biological molecules, there's a special name for that reaction. It's called the dehydration synthesis, or sometimes just dehydration reaction. And if we have two different monomers, one has a hydroxyl on sticking off one end, and the other has a free hydrogen somewhere. Well, what can happen is the bonds can change. So this hydrogen joins this oxygen and forms a water molecule, and then the bond from the first monomer gets linked to that oxygen directly. So the monomers get linked by a covalent bond, so a relatively strong bond that is hard to split apart. And then the opposite reaction can also occur. So we can build polymers by linking these monomers together, or if we have a polymer, we can break it down, like something your stomach might do, or your small intestine as you're digesting your food. You're breaking down these big polymers into their subunits, and in doing that, you're making them available to your body to use for its own purposes. So the opposite of the dehydration reaction is called the hydrolysis reaction. Hydro meaning water, and lysis means splitting apart. So a hydrolysis reaction is one where you essentially add water molecules to this covalent bond in a very particular way, either using enzymes or heat and acidic or basic conditions. And either way, water can get added back to that bond, and we can form the monomers that we started out with. So, or maybe some other organism started out with. Okay, so that's the basic idea. And the way I like to think about it, if you're familiar with the Lego toys, you can, these little building blocks that you can link together, and then you can also break them apart and build something completely different from them. Or maybe you can build the exact same thing if you want. Either way. Okay, so that's the general pattern that we see for these biological molecules. And there are four main classes of biological molecules, and hopefully you've heard of them. These are carbohydrates, lipids, proteins, and nucleic acids. There's a neat little graphic from the Amoeba sisters here that shows the biological molecules and their building blocks, so they're monomers. So you definitely need to know the monomers for each different class of biological molecule. So we're going to start with carbohydrates here. The monomers of a carbohydrate is called a monosaccharide. So that it means single sugar, and they can form either disaccharides, which means like double sugar, or oligosaccharides, meaning a few sugars linked together, or these big long chains of sugar called polysaccharides. And those are things like starch and cellulose and glycogen. But basically, the carbohydrates have a chemical formula that is just carbon and hydrogen and oxygen, and it's in a ratio of 1 to 2 to 1 for these atom molecules. So for each carbon atom, you have basically a molecule of water. So that's how they get their name. And there's more about carbohydrates in the crash course guest lecture video. So I will try not to steal all of that thunder. All right, so lipids. Lipids are kind of weird. They have carbon and hydrogen and a little bit of oxygen, but not very much. So it's mostly hydrocarbons in these lipids. And all of the other types of biological molecules can form infinitely long chains, or well, maybe not infinitely, but arbitrarily long chains. So there's no fixed limit to how long these chains might get. Lipids, on the other hand, can only form 4 different parts. There's just 4 different parts to a lipid polymer. And one of them, we got to wait for the diagram, one of them is a glycerol. And it's got 3 attachment points for fatty acids. So 1 lipid plus 3 fatty acids, or I'm sorry, 1 glycerol plus 3 fatty acids makes up a lipid. And it's a specific type of lipid called a triglyceride, or sometimes a triglycerol. What's the word? Yeah, I think generally triglycerides is probably the best name. I think the other term is triacylglycerol, but that's kind of a tricky one. In any case, these are fats and oils. And there's actually other types of lipids that are mostly hydrocarbons that are all these ring structures like steroids and stuff. But again, the other video will talk about these in more detail. All right, proteins. Proteins are the really, really, really interesting biological molecules because they make stuff happen and serve so many different functions for the cell. So proteins have carbon, hydrogen, and oxygen, and also a little bit of nitrogen, and sometimes a little bit of sulfur. If you've ever been around rotten eggs, that's hydrogen sulfide, which is coming from the decomposition of proteins. And the building blocks of proteins are called amino acids. So those get linked together into these long chains, either called a polypeptide or a protein, and those get folded in a very particular way. Again, I'm not going to say too much more about these because those get covered pretty well. And then lastly, we have the nucleic acids. So the same big 3 atoms, carbon, hydrogen, oxygen. There's also some nitrogen, and there's also some phosphorus. And the building blocks of nucleic acids are called nucleotides. And each nucleotide actually has 3 distinct parts to it, but those get linked together in a very particular way. And these nucleic acids can actually store and relay information, which is really, really cool. So maybe structurally, they might be a little bit simpler than proteins, but the way they interact is really, really interesting. And they actually are a code for building proteins. Oh, did I just give away all of biology? I think I did. Okay, nucleic acids. So again, the monomers are called nucleotides. And each nucleotide is made up of 3 smaller parts. There's a phosphate group, which kind of helps link these nucleotides together. There's a sugar, and the type of sugar changes depending on if we're talking about RNA or DNA. There's just an extra oxygen in the RNA. And then there is a nitrogenous base. So these structures that hang off of the sugars, it's either a single ring or a double ring structure. And these bases are the famous bases A, T, G, and C for adenine, thymine, guanine, and cytosine. Or if we're talking about RNA, there's a different base called U or uracil. And these are basically a way of storing information and also a way of copying information and distributing that information throughout the cell or to offspring cells. And it's really, really cool. And I wish we had time to get into it in Biology 101, but we don't. So if this is really, really interesting to you, you might consider taking Biology 102 or Biology 112 or taking the Majors Biology series. But in any case, those are the monomers. The polymers are these famous molecules, RNA and DNA. So RNA stands for ribonucleic acid. And it's basically usually single-stranded. So we have a single strand of these nucleotides where it goes phosphate, sugar, phosphate, sugar, phosphate, sugar. But then sticking out from those phosphate sugar backbones are these bases that can bind with other things, and they can send signals to maybe build a protein, or maybe help in the building of the protein, or maybe it's like a strand of virus RNA, like from COVID-19, and it's sending instructions to the cell to build, hey, build these proteins, which are actually virus proteins. So RNA is very important in cell communication and the operation of a cell and the building of proteins in a cell. DNA is also a famous molecule, and that stands for deoxyribonucleic acid, because there's one less oxygen atom in the sugar, and the bases are A, T, G, and C, instead of A, U, G, and C, like we'd find in RNA. And DNA is interesting because it likes to form this double helix structure, which is a great, very stable form for the long-term keeping of coded information. So inside of the nucleus of our cells are the chromosomes, which contain DNA and also some protein, but the DNA is kind of the critical thing there. And that DNA can actually get copied when a cell divides. The helix can split apart, and each side can be used to build a new DNA molecule, because only certain bases pair with each other. A binds with T, G binds with C, so every time you have an A on one strand, there's going to be a T on the other. Every time you have a G on one strand, there's going to be a C on the other. And because of this, it's very easy to copy the DNA. And so this is the molecule of heredity. It's what you inherited from your parents, or your biological parents, I should say. And it's what each cell copies and passes on to its daughter cells when they split. It's what can change, and changes in the DNA can cause changes in protein, which is really kind of the underlying mechanism for evolution. Okay, so that is it for my part of this week's lecture. And there's also the additional lecture about carbohydrates and lipids and proteins. So please watch that, and I will see you in class. Stop.